Hello, everyone. Thanks for organizators to call me to here. I'd like to talk about chemical sensing based on carbon quantum dots. I'm professor in physical chemistry in Fortaleza, Ceará. It's a photo of my group of research. I'm a head chief of GCHEMAT, Group of Chemical Advanced Materials. We have some research lines there. Actually, we work with chemistry of materials. We have been working with dielectric and magnetic ceramics, biomaterials. In this case, we work with the mineral phase of bonds. We can get synthesize this kind of materials. We have been working also with biofilms and composites and nanomaterials. In this case, I'd like to highlight these materials because it's the topic of this presentation. Here we can see a quasi-spherical, and we have some layers, graphitic layers. Okay, what is interest in these structures? We have some groups in defects here. We can put a lot of elements like boron, nitrogen, oxygen in with this. We can change the photoluminescence, the yield, the quantum yield. Okay, and we have synthesized these materials less than 10 nanometers. Actually, we synthesize with 2.5 nanometers. And we have some interesting properties. Okay. What is the interest, the story of carbon quantum dots? In this picture, we can see that you can use in biomedicine, optics, photocatalysis, electrochemistry, sensing, and so on. And I highlight the two lines of research. In biomedicine, you can use in photodynamic therapy, bioimaging, and this kind of material is a little bit different the quantum dots, because with quantum dots, we use selenium, cadmium, in this kind of materials is toxic for human. But in this case, carbon quantum dots is not toxic. So you can use to a lot of things, okay? But my topic, it's here, is the sensing. We can make the sense of metal ions, inorganic molecules, for example, pesticides, sugars, food additives. And what a strategy should be used for? The best answer for it depends on what do you want to obtain. For example, you can use the same starting materials to produce a lot of kinds of carbon quantum dots. And this kind of carbon quantum dots can emit in different wavelengths. So you have you take advantage for this, because this material can emit in a lot of wavelengths. From this picture, we can see that we can build a lot of structures, and you have some different properties. For example, you have a lot of colors of these materials, okay? And there are some studies that you can put this in LEDs, in solar cells. So here, I'd like to show a roadmap about my production in this area. Here, we start to produce the first paper with a protein. We use carbon quantum dots to make the sensing of proteins. After, you use to discriminate proteins again, and pesticides in carrot, rice, orange, and so on. In food additives, and now this year we published this paper. I will talk about it in the next slide. It's about a sensor device. So we start here, and now we have set up a device. Okay. In here, next year, you have a chapter book about the sensors by using carbon quantum dots. So the sensing strategy that we can use, it's here. For example, we have a sample with carbon quantum dots, and if you put the analyte, you can analyte, the interaction with the analyte can turn off the light of the carbon dots or turn on. So we have a turn on or turn off sensing, okay? And we have to do this with naked eye. So we have a quenching of this signal. Here is our sensor array. Look here. From this picture, we have the carbon quantum dots and the molecule A, B, C, D. You can see that depends on the molecule, the intensity, the quenching is different. With this, we can create a data bank and use chemometrics tools to like linear discriminant analysis. And with these clusters, when these clusters are well separated, the sensor, the accuracy of the sensor, it's okay. So it's like a fingerprinting. You create a fingerprinting. I'd like to say that it's like a recognition, a face recognition, because the molecules will interact with carbon quantum dots in many ways. But if you want to do this kind of sensor, you need to use with the molecule, the target molecule, and the interference. And why? Why do we need this? Today, the analytical methods to detect and quantify, it's very expensive equipment. You need long time of analysis. The sample preparation is very complex. and you need trained analysts. And imagine if you don't need to use this and use just a small device that you put just a little sample and identify 5, 10, 20 molecules in the same time with this. So you don't need a lab anymore by using this. It's the future, okay? So I put some pictures of the chromatographic equipment that I use, but I hope in future I don't use anymore because it's very expensive. I select 3 papers that I have been published to talk about them and the potential of this carbon quantum dots for sensing. The first one, it was published in the last year, and it's the sensing of food additives based on fluorescent carbon quantum dots. This is my PhD student. He finished, he defends this year, and he is a professor at UFC right now. So, actually, we can synthesize carbon quantum dots with any carbon research. For example, you can use hair, you can use carrots, coffee, everything. Just need to have carbon. So my student is a food engineer, and he use seeds, okay, a natural source. And we select a sample in the supermarket, and here we can see the limit of detection in nanograms per milliliter. You can see that this food additives is used to avoid the degradation of the food. Okay, but I have to highlight one thing here. This setup, with this setup, we can identify five molecules in the same time. Okay, we can see from this picture that the clusters are well defined in the 3 different concentrations. Okay, the next one is the detection and identification of protein using carbon quantum dots. Here we need to change the setup of the methodology. So I believe that it's very easy to see here. We have the carbon quantum dots, protein, and when you mix both, you have a signal here. So we put copper ions to have a second signal, and EDTA. So we can see 3 different informations. So we have, we collected the intensity of the fluorescence intensity of this combination for 8 proteins in the same time. Okay, here we can see the metallic protein and non-metallic protein. So we can do this in the same time with the same sample. Okay, and the last one, it was published this year, two months ago. I like this paper because we can make advantage. We use carbon quantum dots with a nanocomposite, with PVA, to sense nitrate ions emit. So we can see the photoluminescent intensity. You can see here that depends on the concentration of the nitrates on the emit, the signal decrease. It means a quenching of the intensity. Here we can see the nitrate, the difference of nitrate ions and the interference. This interference is the more usually found in the emit. Because of this, we training our sensor to recognize them. So we'll have the anions, the green one, cations, and biomolecules. Okay. This picture is very good because we can see that depends on the concentration of the nitrate ions, the color changes. Here is the device. We use a smartphone, and we bought a pocket microscope. We take this pocket microscope in the internet and put in the cell phone and use a film, a PVA film. So we put the sample in this film, and the film change the color. But it's very difficult to see with the eye. So we use the pixels of this photo, of this sensor. So we need a turn-off sensor here. But to do this, we need to change the surface of the carbon quantum dots. Or carbon quantum dots that we have been synthesized have quantum yield 10%, but we need to make to introduce boron and nitrogen, and we can increase the quantum yield for more than 40%. Okay. Future. I like to dream with this. It's lab on a chip. Actually, today we have lab on a chip, but it's electronic devices. And now in my lab, we have a 3D printer, and we have produced this. And you can see that we have some places that it's like you have all lab on the device in small size. Okay. And what is the advantage of this? You don't need this anymore. You just need a chip in this size, and you put a little bit of your sample, and you can identify 10, 20, 30 molecules in the same time, very fast. Some papers are some workers they are another use for this, the organ on a chip, that you can build an entire organism on this kind of materials. What is the advantage? Fast and cheap diagnostics. Okay. So I'd like to thank the CNPq FUNCAP. We have a collaboration with Chile. And I'd like to thank for all coming.